Hello friends. So today we are going to discuss molecular biology. In this unit we are going to discuss various molecular phenomena. So what we are going to study in this particular unit could be well understood first by going through a central dogma. Central dogma, it is a concept which was given by Watson and Crick. According to the concept of central dogma, the central molecule in any kind of cell is a DNA molecule. That is, all the information for functional or even the structural aspect that resides in a DNA, or in other words we can say that DNA is a genetic material for most of the organism. That is a central molecule. Then according to Watson Crick, the information which is present in the DNA could be copied in a DNA form. This process where the information which is present in the DNA is converted into a DNA form is referred to as replication. So replication literally it means copy of any kind of molecule. If it is a copying of a DNA, the process would be referred to as DNA replication. So DNA replication, it is necessary requirement prior to a cell division, because every newly formed cell must have a complete genetic information. The information which is present in a DNA could also be converted into an RNA language. The information which is present in a DNA could also be converted to an RNA language. And the process where the information which is present in a DNA is converted to an RNA is referred to as transcription. This process is referred to as transcription. So how we can define transcription? Transcription is a process where the information which is present in a DNA is copied in an RNA language. And this information which is now present in RNA, it itself could be utilized for various functional. So RNA itself directly could be utilized for variety of functions. For example, tRNA, they act as an adapter molecule. rRNA, they are the component of ribosomes. We will study also about the siRNA, we will also study about the micron RNA, they have some regulatory roles. And beside this, we will study various other kind of RNA. But if this RNA is a messenger RNA, then the information which is present in this messenger RNA molecule is used as a template for the synthesis of protein. And this process where the information in a messenger RNA is utilized for protein synthesis. This process, as you all know, is referred to as translation. Yes, this is referred to as translation. So how we can define translation? Translation is a process where the information which is present in a messenger RNA molecule could or is utilized for the synthesis of protein. Is it clear? Now moving further. In other words, majority of the function which is present, which is performed by the cell, is performed due to these proteins. So most of the time what happens is the information which is present in DNA is copied in RNA language, and if it is a messenger RNA, then this information which is present in messenger RNA, it is utilized for the synthesis of protein. And these proteins, they perform variety of functions. Let's say they work as an enzyme, they may play an immune functions, they may have some structural roles, they determine the shape of cells. So majority of the functions in a cell is performed by proteins. And this process where the information which is present in RNA is converted to a protein is referred to as translation. As more and more research was being carried out in molecular biology, some additional phenomena they were also being identified. One of the important thing is that it was identified that it is not always necessary that DNA would be the central molecule, because we know there are various viruses which has a RNA as a genetic material. So in some cases we can say that central molecule may not be a DNA, central molecule could be RNA. Okay, if RNA is a central molecule, the information which is present in RNA could also be converted to an RNA form. The information RNA could be copied into an RNA form, as we see in various cases of viruses. If the genome is positive strand, its copied RNA is referred to as negative strand, and if the genome itself is a negative strand, so if some strand which can be directly utilized for a translation process, some RNA strand which can be directly utilized for a translation process, such a strand is referred to as positive strand. And whenever we will make a copy of this from positive strand, we will produce a negative strand. Okay, from positive strand we are going to produce negative strand. But it is not always necessary that the genome would be positive strand. In cases there could be a negative strand, and if the negative strand is a genome, first it is transcribed or copied into a positive strand, and then this positive strand could be utilized for translation purpose. But the important thing over here is not about the negative or positive strand. The important thing over here is that the information which is present in the RNA could also be copied. Although we used this process or name this process also as a transcription, where we will study that, we will say that it is an RNA dependent transcription process. As further research continued, it was found that in some viruses, the information which is present in RNA could be copied into a DNA. This is also possible. And if this happens, this process is referred to as reverse of transcription, or simply referred to as reverse transcription. And this phenomenon of reverse transcription, since it was proposed by Tamin or identified by Tamin, so this phenomenon is also referred to as Taminism. Various retroviruses, including HIV, that is human immune deficiency virus, has an RNA as a genome, and it could be copied into a cDNA, or we can say copy DNA. This process is referred to as reverse transcription. So now what we know is that the information which is present in a DNA could be copied into DNA. The process referred to as replication. The information which is present in a DNA could be converted to an RNA. The process referred to as transcription. The information which is present in messenger RNA could be utilized for the synthesis of protein. The process referred to as translation. Is it clear? And some additional information is also known, that the information which is present in RNA could also be copied in an RNA form. So it would be referred to as RNA based transcription, or the information which is present in RNA could also be converted into DNA. And if it happens, then this process referred to as, this process referred to as reverse transcription, also referred to as Taminism. Up to now, there is no report where the information which is present in a protein could be utilized for the RNA. No such reports are available. Okay. Now, in this particular unit, what we are going to study is, first we are going to study the replication process. Second, with a replication, we are also going to study variety of DNA damage and repair mechanism. Later on in this unit, we are going to study the process of transcription, a variety of RNA molecules and their functions. In next portion of this unit, we are going to study the translation, how protein synthesis occurs and what are the different kind of modifications which may occur in a protein. Beside this, we are also going to study how this replication process is regulated, how this transcription process is regulated in prokaryotes as well as eukaryotes. We are also going to study that how this translation process, it is regulated in a prokaryotes and eukaryotes. So in short, in this molecular biology unit, we are going to study replication, DNA damage, DNA repair, then transcription, regulation at transcription level, then translation and regulation at the translation level, as well as we are also going to focus some post-translational modifications and the most common techniques which are utilized for the process of, or we can say techniques which are routinely utilized in a molecular biology. Is it clear? Okay. So with this basic information, now we are going to start the this unit molecular biology, and first we are going to discuss today the topic. What we are going to discuss is the process of replication. Okay. So first we must define what we mean by replication. Replication could also be referred to as DNA synthesis. This could also be referred to as DNA synthesis. DNA replication, more accurately, we are talking about DNA replication. DNA replication is a process where the information which is present in a DNA is doubled. So we can also use a term DNA duplication. So initially, we would be having one copy of double-stranded DNA. After the process of replication, one round of replication, what we are going to study, what we are going to find is now there would be two copies of double-stranded DNA. Okay. So this is also a double-stranded DNA, and this would be another copy of double-stranded DNA. DNA replication is a prerequisite for a cell to divide. Without duplication of genetic material, a cell cannot divide, because every newly freshly prepared cell, it must have a genetic information. Is it clear? So prior to a cell division, what important requirement? The important requirement is the DNA must be duplicated so that it could be equally distributed among the two cells. Okay. So where, or more accurately, when this process of DNA replication occurs? Okay. So what we are saying is that stage where this DNA replication occurs, means when actually this DNA replication occurs. According to bacterial cell cycle model, in bacteria, we study that there are three stages: I, then there is a C stage, and then there is a D stage. I could be referred to as interval. I is referred to as interval. C, where there is a chromosomal replication, and D is the division, where the bacteria divide. So in a bacteria, we can say that the C stage is the stage of cell cycle where the DNA is synthesized or genetic material is duplicated. Is it clear? During a chromosomal replication or during a C stage. If we talk about the eukaryotes, including humans, plants and fungus, if we talk about the eukaryotes, in eukaryotes, the cell cycle, it comprises of G1 stage, S stage, G2 stage, and then there is a M phase. In case of eukaryotes, basically the DNA replications occur during the S phase. So S phase of cell cycle, this is the stage where there is a DNA replication. Is it clear? So first thing which should be kept in the mind is that in bacteria, the DNA replicate during the C stage of cell cycle. In eukaryotes, the DNA replicates during the S stage of cell cycle. Is it clear? Okay. Now coming to some basic properties of DNA replication, which is common both in prokaryotes as well as eukaryotes. Okay. So some common. First important thing is that DNA replication, as you all know, DNA replication is semi-conservative process. DNA replication is semi-conservative process. What we mean by this is that during DNA replication, let's say this is some parental DNA strand, a double-stranded DNA, and after the DNA replication, what we find is that from one double-stranded DNA, we get two double-stranded DNA molecule. But what we find is that one of the strand is parental and another strand is new. So the strand which is drawn in a blue color is a new strand, while the strand which is black in color is a parental strand. So this process where one of the strand is new while another strand is parental, the process referred to as semi-conservative. This could be explained further that if, let's say, if this would have been a case that this is some double-stranded DNA, and what we find is that we get a same kind of double-stranded DNA and another DNA strand which is totally different type. If this is a case, so here both the strands are parental and both the strands they are new. If this is a case, or we can say if this would have been a case, we will say that DNA replication is conservative. Okay. If the process like that, this is the parental strands, one of the strand is parental while another strand is new, the process is referred to as already mentioned, the process would be referred to as, yes, the process would be referred to as semi-conservative. And a third scenario is possible that if suppose these are both the parental strands, and what you find is that this nucleotides which is present in this DNA, they get degraded, and some new nucleotide and the old nucleotides, they mix together and form the DNA, somewhat like this. The original DNA is degraded into nucleotides, and then these new nucleotides and old nucleotides, they get mixed together and form a two DNA strands. Then the process would be referred to as dispersive, or we can also name it as a non-conserved. So both the strands, they are synthesized from the older nucleotides and new nucleotides, and they mix together. This would be referred to as dispersive. One strand is completely parental, another strand is completely new, semi-conservative. Another third scenario, or we can say this, in this scenario, one of the DNA has both the parental strand and another double-stranded DNA is having both the new strands, the process referred to as conservative. Out of these three different modes of replication, it was found that in biological system, the DNA replication is semi-conservative. DNA replication is a semi-conservative. This was first time proven by the Michelson and Stoll. So it was proven by Michelson and Stoll in Michelson and Stoll. Okay. Michelson and Stoll, they proved in E. coli that DNA replication is semi-conservative. So coming to the point that how actually they proved that DNA replication is semi-conservative. For this, what they did is they maintained the E. coli for various generation on a N15, that is a heavier isotope of nitrogen, N15 containing medium for various generations. So for multiple generations, the E. coli were being maintained on N15 containing medium. So let's say this is some petri dish where you are going to culture these E. coli, and these E. coli, whatever they are growing, they were being maintained on a medium containing an N15. And when the bacterial colonies, they were isolated here, and then they were centrifuged using a bond density centrifuge. What was identified is that the isolated DNA, it is forming a one sharp band at a higher density, which represents that here both the strands, both the DNA strands, they are of heavy type. We can say that both the strands, they contain a N15 type of isotopes of nitrogen. So we can, for N15, we can also use the term heavy. N15 is a heavier isotope. This is the bond density centrifugation tube, where there is a low density and there is a high density on this side. So the DNA is being separated according to the density. There is a one sharp band which suggests that all the bacteria, they are having only one type of isotope of nitrogen, and that is N15. Is it clear? And then what they did is they transferred these bacteria to another medium which was containing an N14. So N14 containing medium, and they placed it for a one generation. Okay. Now again, various bacterial colonies, let's say they could be various bacterial colonies. Now they were being maintained on N14 containing medium. DNA was isolated from them, and again they were analyzed by bond density centrifugation. And what they observed is that again there is a single band, but comparatively at a higher position. Okay. This was the original position. The DNA band is not at original position. It is at certain, we can say, intermediate density, which suggests that here definitely the DNA replication is not conservative. If DNA replication would have been conservative, then you must get the bands at exactly same place, and another new strand could be at another place which should be of very low density. Similarly, but this intermediate density may be due to two different cases, because okay, we compare. If it would have been conservative, then this should be the band at a higher place. You must get one band at this place, which include this both heavy strands, and this should be another band which should be somewhere here. Okay, there should be another band somewhere here, which must represent new strands. We can also name it as light. But this was not a case. You were getting a one band, and you can get one band either in this case. In this case, you will get a one band of an intermediate density, or you will get a one band even in this case. So to rule out that which condition is the actual case, whether the DNA replication is semi-conservative or whether the DNA replication is a dispersive, what they did is that for one more generation, they placed the bacteria on an N14 containing medium. They placed once more on an N14 containing medium. Now, what they observed? Again, bacterial colonies were being maintained on this N14 containing medium. Let's say again, the various bacterial colonies were there. DNA was isolated, analyzed through the bond density centrifugation. Again, they were analyzed by bond density centrifugation. What they observed is that now they are getting two bands, one exactly at the same place, another at a higher place. So this is the case. This was the exact same place, and now you are getting one more band at this place. This basically rule out that DNA replication is dispersive. DNA replication is now not dispersive. The DNA replication, now there is only one possibility that this could be observed if only the DNA replication is semi-conservative. Yes. Because if the DNA replication is semi-dispersive type, then what should happen is that again this DNA must be degraded, some old and new nucleotides, they must be synthesized. Again you should get a one band, but you are getting a two bands. So conservative was ruled out on transfer to N14 medium for a one generation. This case was also ruled out when subsequently the bacteria were transferred on N14 medium for a second generation. So finally they concluded that DNA replication is a semi-conservative. So let's apply your mind and try give me the answer. What could be the answer? Let's say if let's say the same experiment was performed by you for some unknown bacteria you captured from a Mars and you obtained this kind of result, what prediction you will make? So first what you did is that you collected some bacterial species from a Mars, let's say from another planet, and you maintained it on an N15 containing medium, and then for a first generation and then for a second generation you are placing them on an N14 containing medium. Let's say these are representing the densities, okay, and this is the density from a lower dense to a higher side. When you place these bacteria on N15 containing medium, you was getting a band at this place, okay. Then you transfer these bacteria that you isolated from. Suppose a hypothetical question is there. Suppose you isolated a bacteria from Mars, and again you are placing them on a medium containing N14 for one generation, and surprisingly you get again a band at this place as well as another band at this place. And further, when you place it on N14 medium, again you are getting one band over this place and another band somewhere here, and almost of double width. What conclusion could be made? Now can we say that DNA replication is semi-conservative? Apply your mind. No? Yes. Answer is no. Now we can say that DNA replication is conservative. Why conservative? Because let's say that these are the two DNA strands, double strand DNA. Both are of heavy, and I am drawing these heavy strand by blue color. If DNA replication is conservative, then these bands should appear from one generation to next generation. They should always appear. This DNA should always appear. This DNA should be always there. It should remain conserved, and whatever the DNA which is being synthesized, this should be of new type. It should be like this. So you are getting one band of this, another band of this. Now after one generation there would be two DNA strands, and after second generation there should be four DNA strands. It means all of them they should be of these green type. So that's why the width of this band should be comparatively very large as compared to this band. The width here is exactly same. So if this is the case, then we will see that DNA replication is not semi-conservative. DNA replication is conservative. And let's say again hypothetical question is that if you get a band pattern like this, if you get a band pattern somewhat like this, then if this is the case, you will say that DNA replication is conservative, okay. Now let's say if condition would have been somewhat like this, if the condition is somewhat like this, then how your predictions will get changed. You get one band over here, another band over here, and another band again over here, almost at the same place. Now, is DNA replication semi-conservative? No. In this case, we will say that DNA replication is dispersive. DNA replication is dispersive. First you get a band at higher place, then at intermediate density, again at almost intermediate density, or slightly we can say higher than that. In semi-conservative, what you are getting, in case of semi-conservative, what you get is initially when you transfer it on an N15 containing medium, you get a one band at a higher density. Then in first generation on to transfer to N14 medium, you get one band at a intermediate density. And on second generation on transfer on N14 medium, you get now two bands. So first you get one band over here and then two bands over here. So this is on N15 medium, this is on N14, this is also on N14 containing medium. This is for first generation and this is for the second generation. And if you keep this on even for a third generation, now what you'll find is that the DNA which of intermediate density will remain same. Majority of the DNA which would be synthesized will be of lower density. So this would be after the third generation, when there were 2 DNA strands. From 2 now there are 4 DNA strands. So you can say that now this is representing a heavy light strands, and rest of them they are referred to as light strands. Okay, is it clear? This is semi-conservative. So let me ask you one more question. Let's say you are having both heavy strands, and then you transfer it to, for first generation on N14 containing medium, for a second generation also on N14 containing medium, and for a third generation on N14 containing medium. What proportion of the DNA strands would be a heavy type, or heavy light type, or light type? So we are allowing them to multiply for 3 generations. Initially they were being placed on heavy type. So after 3 rounds, what would be the proportion of heavy, what would be the proportion of heavy light, and what would be the proportion of light? Okay, you got the question? Then try to solve this. Okay, I'm giving you 1 minute. Apply your mind. Okay, now let me solve it for you. So original strand is heavy, and you know that this heavy strand will open up for the first generation, both the heavy, assuming the DNA replication semi-conservative. This would be after the first generation. You are putting it on N14, so both new strands would be light. Okay, so after first generation you will get a 1 band, and both the DNA strands would be heavy light, that would be of intermediate density. During the second generation, what you are going to observe is this will open up heavy strand, light strand. This will also open up heavy strands and light strand. And again for second generation you are giving a N14. So there would be light strand over here, light strand over here, light strand over here, light strand over here. So after second generation you are getting a 2 heavy light strand and 2 light strand. Is it clear? Okay. What is going to happen during the third generation? So again this heavy and light will open up. This light and light will open up. This heavy and light open up, and this light and light will open up. They would be used as a template for the synthesis of a new round of DNA. And again for third generation you are providing a N14. So again the light strand would be there. Okay, light strand would be there. So now after 3 rounds you will have a 8 double-stranded DNA molecules. Okay, you are having 8 double-stranded DNA molecules. That's confirmed that you will get a 8 double-stranded DNA molecules. After first round you will have 2 double-stranded molecules. After second round you will have 4 double-stranded DNA molecules, and after third round you will have a 8 double-stranded DNA molecules. So what would be the proportion of heavy? The proportion of heavy you can see is 0. The proportion of heavy light, is, this is 1 heavy light, this is another heavy light, is 2. And rest of them would be light. 1, 2, 3, 4, 5 and 6. These would be heavy light. So you can say that proportion would be heavy light and light. So do try your hand on this question. I am giving one more question. Let's say you are having some certain bacteria, let's say E. coli, where the DNA replication is semi-conservative, and you are having you are culturing them on an N15 containing medium. They were originally maintained on N15 containing medium, and it was being said that for first generation they were transferred, first and second generations they were transferred on N14 containing medium, and for third generations they were being maintained on again they were provided with an N15 containing medium. In this scenario, try to find out what would be the heavy strands, what would be the number of heavy light strands, and what would be the number of light strands. I can say you one thing, that since there are 3 generations, so total DNA molecules which you are going to get is 8. Now simply you have to identify that what will be the number of heavy, what will be the number of heavy light or light strand. Is it clear? This is question. You apply your mind. Okay. I can simply say that the answer what you are going to get would be somewhere you will get this, you will get 0, you will get this as a 4, as well as this would be also 4. So try, you will get some answer like this. Okay. So it was Messelson and Stoll who excellently proved that DNA replication is semi-conservative in E. coli. The same thing could also be proven using we can say bromouracil. Bromouracil is basically, we can say, a nucleotide analog for thymine. In eukaryotes, it could be proven that, let's say, there is some eukaryotic chromosome. Okay, and this is, let's say, a eukaryotic chromosome. These are the 2 chromatids linked at the centromere. Is it clear? And we know that these each chromatid has a 1 double-stranded DNA molecule. So for our simplicity, we are now drawing this like this. This is the case, that each chromatid is having a double-stranded DNA molecule. This is present in this is present in this. Normally, in a DNA, you will find that there are adenine, thymine, cytosine, and guanine. Now, if you give, let's say, put this on a bromouracil for 1 generation. Bromouracil, as I have said, it is an analog of thymine. So instead of thymine, you are giving a bromouracil. And bromouracil could be easily detected using an antibody raised against a bromouracil. Antibodies, they could be tagged with a fluorescent dye. They can recognize the chromatid having a bromouracil. They will show a fluorescence. Is it clear? So if DNA replication is semi-conservative, what should happen is that out of, so let's say bromouracil, we give it as a blue color. If DNA replication is a semi-conservative, then what is expected is that now one of the strand, this must open up, should act as a template, and the new strand during synthesis, instead of thymine, it must incorporate a bromouracil. This should be the case. So let's say from this chromatid, from 1 chromatid, again these 2 chromatids, they are being synthesized. From this also 2 chromatids would be synthesized. We are simply at present focusing on this particular region. Okay, that is, now this single chromatid, it has been copied into again 2, and what you will find is that now both the chromatids of will incorporate a bromouracil. One of the strand will contain a bromouracil, and one of the strand is the parental strand. You will get 50% of, you can say 50% incorporation of bromouracil in both the strands. If DNA replication is semi-conservative, then also only it is possible that both the chromatids, now they will show an incorporation of bromouracil. And if you put it for one more generation on a bromouracil containing medium, so let's say this is again replicated for next round, what should happen is that, let's say now this is blue colored, which is acting as a template. This is a black color, which is also acting as a template. And you are providing again a bromouracil. Bromouracil we are marking to the blue. You will get one double-stranded DNA, which would be completely of blue color. Another DNA, double-stranded DNA, where one of the strand would be having normal thymine and another strand would be having bromouracil. That is, now you will find that there should be one chromatid where the complete both, or we can say where both the strands must be containing a bromouracil. In another strand, in another chromatid, one of the strand is having a bromouracil while another is having a thymine. If this is the case, we can say that DNA replication is semi-conservative. So even in eukaryotic system, using a bromouracil, it was identified that DNA replication is a semi-conservative. So bromouracil is simply an analog. So instead of using the heaviest of, here we are using a term bromouracil. So we can say this is a normal thymine strands, now thymine bromouracil strands. Thymine bromouracil strand. Now you're getting a bromouracil strand over here, bromouracil thymine strand, and this is possible only if the DNA replication is semi-conservative. Okay, so in both the cases, that is in a prokaryotes as well as eukaryotes, it was identified that DNA replication is semi-conservative. Okay, now moving further. DNA replication is a template dependent process. DNA replication is template dependent process. That is, we must have some existing information which must be copied, or one strand or one DNA strand must act as a template for copy of new strand. So DNA replication is a template dependent process. In other words, we can say that the template strand is in 3' to 5' direction, while the replication basically it occurs in 5' to 3' direction. That is, there must be some kind of template, and template is 3' to 5', which is having some kind of, we can say, nucleotide, and simply having a template is not sufficient. Template is basically the single-stranded DNA, but having a single-stranded DNA is not sufficient. Besides single-stranded template, we also need a primer. So more accurately, we can say that template primer junction is important. It's somewhat like this is available. Then further DNA could be replicated. The replication direction, it would be 5' to 3'. So this is the replication. So what we can say that for replication process to occur, there is a need of first template. You need a template which is 3' to 5'. You need a primer, and then this primer could be further extended. Without template, replication or DNA replication will not occur. Even there is a single-stranded template, but there is no primer, or we can say there is no free 3' end, DNA cannot be replicated. So why the template strand is 3' to 5'? Because the replication is 5' to 3' direction. That is, the DNA is double-stranded having an anti-parallel strands. DNA is double-stranded, both the strands are anti-parallel. So in this scenario, definitely template strand and replicated strands must have opposite polarity. Replicated strand must have opposite polarity. If template strand is 3' to 5', the strand which is synthesized must be 5' to 3'. Okay. So the important thing is that what we need is, we need template, primer, junction. it is available, then the DNA could be replicated in majority of cases. So let's say I'm giving you some cases where you believe that DNA replication could be carried out. Let's say this is some kind of double stranded DNA. Here this is a single stranded DNA which is circular. This is single stranded DNA linear form, and whenever we represent DNA in a linear form, its orientation or polarity must be given. Similarly, you are having a circular DNA molecule with some short DNA added to it. You are having one DNA molecule somewhat like this. This is, let's say, 5' to 3', and this is 5', this is 3', and let's say this is another scenario represent like this. This is, let's say, 5', 3', 5', 3'. This is double stranded over here. Now you have to give me an answer that out of these 5 scenarios where the replication is possible if you add a DNA polymerase, where DNA polymerase is able to extend or it is able to synthesize the DNA. Apply your mind. These are the different cases A, B, C, D, E and F, where if you what you are adding is you are adding only DNA polymerase. Okay, you are adding a DNA polymerase where the replication will proceed. So let's analyze one by one. In case 1, DNA polymerase will not able to replicate because the DNA is not single stranded. First you need a helicase, that will study later on, but if it is a double stranded, DNA cannot be replicated. Here the DNA is single stranded, but there is no free 3' end where a DNA polymerase can extend. Here also a single stranded DNA template is available, but the replication cannot be continued because DNA polymerase need a free 3' OH for an extension. Here replication is possible because there is a single strand template as well as 3' OH which could be utilized and which could be extended. DNA could be synthesized. Here you will observe a DNA synthesis. In this case also you will observe a DNA synthesis because the free 3' end is available and a template is also there. Synthesis would be observed even in case of D. In case of F, 3' end is available but there is no template in front of this, so it cannot be extended. 3' end is there because DNA replication cannot occur or it cannot extend 5'. DNA polymerase will always extend a 3' end. So here also replication is not possible. So we can say in a case C and D, if you add a DNA polymerase, DNA could be extended because here both the scenario is being fulfilled, that you are having a single stranded template and you are having a short oligonucleotide primer having a 3'. It doesn't matter whether it is short or long, but it must have a free 3' OH group which could be extended. Is it clear? So the important point is the DNA replication is template dependent process. DNA replication occurs in a direction of 5' to 3'. DNA replication template strand is 3' to 5'. So DNA replication is a template dependent, and most of time the template is the DNA and the DNA polymerase which uses DNA as a template are referred to as DNA dependent DNA polymerase. In some cases what you will find is that the role of template could be performed by an RNA. RNA could also act as a template. RNA could be utilized as a template for the synthesis of DNA. The DNA polymerases would be referred to as RNA dependent DNA polymerase. And you can give any example of RNA dependent DNA polymerase. Do you have any example? Yes, there is one example you all might be knowing is reverse transcriptase. So reverse transcriptase is basically, you can say that it is RNA dependent DNA polymerase. Even you will study that telomerase enzyme, which basically replicates the ends of we can say chromosomes in higher organisms. This telomerase is also an RNA dependent DNA polymerase. Majority of DNA polymerase, what we are going to study in prokaryotes and eukaryotes, like DNA polymerase 1, 2, 3 or DNA polymerase alpha, beta, gamma, delta, epsilon, etc. They all are DNA dependent DNA polymerase. But there are some exceptional DNA polymerases which uses RNA as a template, and two such examples include, one is reverse transcriptase and another is telomerase. Is it clear? So the important thing is DNA replication is a template dependent process, and not only template dependent process, there is a need of a primer which provides a 3' OH. In other words, this could be further extended in this, elaborated in this point, that DNA replication occurs in 5' to 3' direction. If template is 3' to 5', then replication is in 5' to 3' direction. What this means is that the DNA polymerase, it extends. Let's say this is a strand. Let's say this is some template strand. These are some of the nucleotides in a template strand. And let's say the template strand has a polarity 3' to 5'. And let's say this is some kind of already synthesized DNA or some RNA which is acting as a primer. This is 5' and this is 3'. Okay. The new nucleotides, they are added onto a 3' OH group of existing nucleotides. Means if some nucleotides is being added, it is being added over here. So again, it's 5' end will get linked and 3' end would be free. So it will get linked and again 3' end would be free. So what we are saying is that replication is occurring in a direction 5' to 3' direction. What this means is that here we can write that DNA polymerase adds new nucleotide. They adds a new nucleotide to 3' OH of existing polynucleotide chain. That is 3' end is, this would be further extended. So that's why we see that DNA replication occurs in a direction 5' to 3'. We can move a little bit more closer to the chemistry of this polymerization. Now what happens is that, let's say this is some kind of template strand which is having an adenine, this is having a thymine, let's say this may be a cytosine, this may be a guanine, this may be adenine, and so on. And this is template strand, means this is 3' to 5', and this is some nucleotides which is, let's say, already added, which will serve as a primer 5', and here there is another nucleotide, that is adenine. And we are saying that 3' OH group is free. 3' OH group is free. For replication, what we need is, we need a deoxyribonucleotides. We need a deoxyribonucleotide means we need a D-NTP. So there are different D-NTPs they are available. Let's say there is some nucleotide, let's say guanine, having a 3-phosphate. It is there. These 3 phosphates, as we have studied in biochemistry, it is alpha, beta, and gamma phosphate. First, this enters into the active site of DNA polymerase. So there would be some enzyme over here which is working, and this enzyme which is going to work over here would be referred to as DNA polymerase. Let's say we are drawing a rough diagram like this, that there is some kind of DNA polymerase which is working over here. First, the guanine nucleotide, it will enter into the active site. It is a deoxyribonucleotide which is going to enter over here. When it enter over here, this 3' OH group, it is going to attack on a beta and gamma phosphate. This pyrophosphate would be released, that is beta and gamma phosphate would be released, while this alpha phosphate, it will become a part of phosphodiester bond, and one more nucleotide will incorporate over here, that would be guanine, whose again 5' end has linked, but its 3' end would be again free. So this nucleotide, it get enter over here, releasing a pyrophosphate over here. Is it clear? So what we are saying is that for synthesis, what we need is DNTP. Only DNMP, that is nucleoside monophosphate, will become a part, while a pyrophosphate would be released. This energy which is released during this, because whenever this is an energy rich bond which is being cleaved, whenever this energy rich bond would be cleaved, it will provide an energy for, it is going to provide you an energy for this DNA synthesis. Energy rich bond which is between alpha and beta phosphate, this is basically utilized for the synthesis process, and pyrophosphate is released. And further, to stop the reversible reaction, this pyrophosphate is cleaved into 2 inorganic phosphate by another enzyme referred to as pyrophosphatase. Is it clear? So first reaction is carried out by DNA polymerase. So what here I want to mention is that whenever a deoxynucleoside triphosphate, it become a part of DNA, one energy rich bond is hydrolyzed, which is between alpha phosphate and beta phosphate, which provide an energy for DNA synthesis. During this time, pyrophosphate is released. This pyrophosphate is further cleaved by pyrophosphatase, so that to inhibit the reversible reaction, so that this added nucleotide should not again get excised from the 3' end. Is it clear? Okay. So that's why we say that DNA replication occurs in a 5' to 3' direction. So question may be asked, from where you get an energy for the DNA replication process? The energy comes due to an energy rich bond which is between an alpha phosphate and beta phosphate. Another question is that, suppose if you want to radio label the newly synthesized DNA, which phosphate should be radioactive? Alpha phosphate, beta phosphate, or gamma phosphate? Which phosphate should be radioactive? This phosphate should be radioactive, or this phosphate should be radioactive, or this phosphate should be radioactive? Apply your mind, means what you should provide. Alpha 32 labeled alpha 32, we can say more accurately, alpha phosphate 32 labeled ATP, this will work, or more accurately DATP. Beta phosphate 32 deoxy ATP, that would be utilized, or gamma phosphate 32 DATP would be should be utilized. Out of this, according to this, what should be utilized? Or one more choice, alpha P32 labeled ATP, that must be utilized. So first thing is that ATP must not be utilized, because in DNA, instead of normal nucleotides, deoxyribonucleotides are utilized. And if you want to radio label the strands, alpha phosphate, beta phosphate, and gamma phosphate, what you find that only alpha phosphate is going to become a part of this polynucleotide chain. Beta phosphate and gamma phosphate, they are released. So beta and gamma phosphate, if they are radioactive, they will not become a part. Beta and gamma phosphate, they are being released. Only if there is an alpha phosphate, it will become a part of existing DNA chain. Is it clear? So answer would be, you must take this kind of deoxy ATP so that the DNA strands can get radio labeled. Although now the most common way is to utilize tritium hydrogen labeled thymine, because it would be specifically incorporated into DNA, because taking a radioactive phosphate nucleotides, phosphorus phosphate is comparatively strong emitter, and second, almost all the phosphorus bonds, they will get radio labeled, and it will make the DNA more unstable. Then that was the important point, that DNA replication is occurring in a 5' to 3' direction. Template is 3' to 5'. Another important thing is that DNA replication is semi-discontinuous process. DNA replication is a semi-discontinuous process. And what we mean by this? We have said that DNA replication is a conservative process, semi-conservative process. DNA replication is a template dependent process. DNA replication occurs in a 5' to 3' direction. Then we are saying that DNA replication is a semi-discontinuous process. From this, what we mean that out of the two DNA strands, so let's say this is some DNA, both are the parental strands. Let's say this is a 5', this is 3', this is 3' template and this is 5' template. What you find is that one of the strand is being continuously synthesized. One of the strand is being continuously synthesized. And the strand which is being continuously synthesized is referred to as leading strand. This is referred to as leading strand. If another strand would have also been synthesized like this in a continuous manner, then we will see that DNA replication is a continuous process, but this is not being observed. Both the strands, they are not being continuously synthesized. What is being observed that another strand, it is synthesized in a short patches like this. It is replicated in these short patches. This strands which is synthesized in a short patches is referred to as lagging strand. This is referred to as lagging strand. Although the direction of every strand which is being synthesized is a 5' to 3', this is also 5' to 3' strands. So one strand is continuous, another strand is discontinuous. That's why this overall process is referred to as semi-discontinuous process. And why this process is semi-discontinuous? Why both the strands, they are not continuously synthesized? The answer is again the same, because replication occurs in 5' to 3' direction. That is, if you are trying to say that DNA replication in this strand is also continuous, then what you are saying is that DNA replication is occurring in a direction of 3' to 5', which is not possible. Here continuous synthesis is possible because this is occurring in a 5' to 3' direction. Here this is synthesized in a short stretches in a direction of 5' to 3' direction. This would be more clear as we will study later on the process of replication. Is it clear? So DNA replication is semi-discontinuous process. One of the strand is continuously synthesized, another strand is discontinuously synthesized. The process referred to as semi-discontinuous. And these short fragments which are synthesized on a lagging strands, these are referred to as Okazaki fragments. These short fragments are referred to as Okazaki fragments, which in prokaryotes could be comparatively very large. In prokaryotes, like E. coli, these could be around 1,000 to even 2,000 nucleotide. While in case of eukaryotes, they are comparatively much smaller. They are often around 100 to 400 nucleotides. So what we can say, we can take a point from here is that Okazaki fragments, they are shorter in eukaryotes. They are ranging from 100 to 400 nucleotides, while Okazaki fragments in case of prokaryotes, they are comparatively longer. They ranges from 1,000 to 2,000 nucleotides. Is it clear? So let's move to another point. So these are some of the common properties between prokaryotes and eukaryotes, that DNA replication is semi-conservative. DNA replication occurs in 5' to 3' direction. The template is 3' to 5'. DNA replication is a semi-discontinuous process. Now, DNA replication in prokaryotes, what you find is in prokaryotes, the DNA replication is most often it is single origin bidirectional. While in case of eukaryotes, the replication is multiple origin bidirectional, because eukaryotes have comparatively longer genome size as compared to bacteria. So instead of starting the replication from one place, the replication is simultaneously started from multiple places. So we can say like this: in a bacteria, the replication is it is started from a single place and moving in both the directions. So let's say this is how the DNA is being synthesized in case of bacteria. This is the place from where the replication has been initiated. You can say that this is the place from where the replication has been initiated. Well, instead of this, in case of eukaryotes, higher organisms including yeast and human, what you find is that replication is initiated from multiple places, and from each places the replication is moving in both the direction. Is it clear? So replication is starting from these origins and moving to both the directions. So multiple origin, bidirectional. From every origin the replication is proceeding in. Here there was a single origin and bidirectional. So single origin and multiple origin. It is not always a case that bacteria will have a single origin. There are various known cases of bacteria where there may be 2 or even 3 different origins. But majority of bacteria, including E. coli, has only single origin. And again, it is not also necessary that always a genome of a prokaryote would be circular. In some cases, even the genome of bacteria could be linear, even it could be fragmented. Well, in case of eukaryotes, what we find is that the genome is most often linear double-stranded molecule. Then, so mostly we are saying that DNA replication prokaryotes is single origin, bidirectional, while in case of eukaryotes it is multiple origin, bidirectional. With this basic introduction, now we are moving to various proteins which are being utilized for the replication process. We are going through the proteins one by one which are basically utilized for the replication process. These places from where replication starts in bacteria is referred to as ORIC. In case of we can say yeast, they are referred to as ORIC. In case of E. coli, from where the replication is initiated, and yeast, from where multiple places from where these replication started, is referred to as autonomous replicating sequence. We will study in detail about these ORIC or ARS sequences. But one thing which is common in both these cases is that both are 80-rich sequences. That is, whether it is a ORIC or it is a ARS. The common thing is that both are 80-rich. And benefit of having a ARS sequence is of 80-rich sequence is that they can be easily opened up during the replication process. It is comparatively easier to open these kind of sequences because A and T, they are having only 2 hydrogen bond, while G and C, they are having a 3 hydrogen bond. Is it clear? So with this, now we are talking about various proteins, or we can say enzymes, which are used for replication, which are involved in the replication process. So let's have a quick view on these proteins. The first proteins which are utilized for the replications are initiator proteins. Proteins are those proteins which basically recognize ORIC in case of bacteria, ORIC of E. coli. And the role of this initial protein in E. coli, it is performed by a protein referred to as DNA A. This is a initial protein. So we will study later on that DNA A, when it is in ATP bound form, it recognizes a ORIC. The same initial protein, it basically in case of yeast, it recognizes a ARS, autonomous replicating sequence in a yeast. And the name of a initiator protein, which is recognize the ARS in yeast, is referred to as, simply the name is referred to as ORC. That is origin recognition complex, ORC. So initiator protein in case of eukaryote is ORC. ORC stands for origin recognition complex. So these are the, we can say, first protein, whether it is a bacteria or it is eukaryote, these we can say are the first proteins which are going to recognize whether it is a eukaryote or prokaryote or a eukaryote. And let's say these are the places from where the replication could be started. The first protein which is going to bind over here, which is having an ability to interact with a DNA, is an initiator protein. That could be either DNA A, or a protein which is going to bind on these places is a ORC. ORC or origin recognition complex, it is most often hexameric protein composed of protein referred to as ORC 1 to 6. That is, there are 6 subunits, namely ORC1, ORC2, ORC3, ORC4, ORC5 and ORC6. Okay. So first protein which is required is a initiator protein. So let's say here initiator protein named as ORC bind to this, or a DNA A protein binds to this. This is a DNA protein interaction. The second category of protein which is required is basically the helicase. And as the name suggests, helicase, it disrupts helical structure. It disrupts helical structure. It disrupts helical structure. What we mean from this? That they break hydrogen bond. It means what they do is they convert a double-stranded DNA into a single-stranded DNA. So let's say this is a double-stranded DNA, and this double-stranded DNA from some particular localized region, it is converted into a single-stranded by the enzymes referred to as helicase. So there are some enzymes which, let's say, work over here and converting this DNA into a single strand form, so that replication could be initiated. Okay, let's say here now the DNA is in a single strand state. You are getting some kind of replication bubble. Here the complete DNA was double stranded. So some particular region of the DNA from a double stranded state is converted to a single stranded state by an enzyme referred to as helicase. For this helicase, they require an energy, and the energy is utilized in form of ATP. So helicase, to disrupt a hydrogen bond, they need an energy, and this energy is derived from, it is derived from ATP. The role of helicase in case of the role of helicase in a prokaryotes is found by, it is by a DNA B protein. DNA B protein, it acts as a helicase. In case of eukaryotes, let's say from prokaryotes, whatever we are taking an example is from E. coli, and in eukaryotes, whatever example we are taking is from the yeast. In case of eukaryotes, the role of helicase is performed by MCM protein. MCM, again, it is a hexameric protein composed of 6 subunits referred to as MCM 2 to 7. So the helicase, it is known by different name in bacteria and eukaryotes. In bacteria it is referred to as DNA B, and in eukaryote it is referred to as MCM227. Both of them, they are hexameric protein. Hexameric protein means they are composed of, you can say in this way, we can say that they are composed of 6 subunits. So they are hexameric protein composed of almost 6 subunits. Whether it is a DNA B protein or whether it is an MCM227 proteins, and they need an ATP for that particular functioning. One more important point what I want to make over here is that helicase, they also has a polarity. That is, the helicase can migrate in a 5' to 3' direction, or helicase they can migrate in a 3' to 5' direction. Most of the helicase which work on, we can say they use a lagging strand. Most of the helicase, they use lagging strand as template. Whenever they are using lagging strand as a template, what you'll find is that they will migrate in helicase, they migrate in a 5' to 3' direction. What I want to say over here is, let's say that this is, let's say some DNA strand, okay, and we are talking only one half, a very small portion, you can say we are talking about a very small portion. Let's say this is the 3' strand and this is a 5' strand, and as already mentioned that this strand would be continuously synthesized, and this strand would be referred to as leading strand, and this strand would be discontinuously synthesized, okay, this would be referred to as lagging strand. So if this is, we can say that template for lagging strand, and if a helicase, because helicase definitely it has continuously, it has to move in this particular direction, and if helicase it is using this lagging strand template, then it has to move in which direction? You can see it is moving in a direction 5' to 3'. So more rapidly what we say that when lagging strand template is used by helicase move in a direction from 5' to 3'. Is it clear? So majority of the helicase, they migrate in a 5' to 3' direction, because they use that particular strand which is the template for lagging strand. If they are using this as a riding strand for movement, then they will migrate in a 5' to 3' direction. Let's say if there would have been another scenario, because this is one thing very clear, that helicase it has to move in this particular direction, they have to move in this particular direction. But now if they are using the strand, now let's say if they are using the strand 3' to 5' for the movement, in other words, this strand would be referred to as continuous strand, this would be referred to as discontinuous or lagging strand. And if helicase is moving on the strand, if a helicase it is moving on the strand which is being utilized as a template for a leading strand, under this scenario what you will observe is the helicase is moving in a direction 3' to, it is moving in the direction 3' to 5'. So both kind of helicase, they are all available. The helicase can move in the direction 5' to 3', or helicase they can move in a 3' to 5', depending upon which out of these two strands. This strand could also be used, this strand could also be utilized for movement, or this strand could be also utilized for the movement. Now it depends, helicase out of these two strands, which strand is being utilized for the movement. It could be in a, if the strand which is a template for leading strand is being used for movement, then movement would be in a 3' to 5' direction. And if the strand which is a template for lagging strand is utilized by a helicase, then the helicase movement would be in a 5' to 3' direction. So this is very important point. Then moving further, that helicase, they need an energy in form ATP, as I have already mentioned. Then another protein, a third protein which is required for the replication to continue, are referred to as single stranded binding protein, in short referred to as SSB, or in a prokaryotes they are simply referred to as SSB, that is single strand binding protein. In case of eukaryotes, they are referred to as replication protein A. The function of these SSB, or we can say replication protein A, is to they keep DNA in single stranded state at replication bubble. Until replication machinery, or more accurately until we can say replicative polymerase, until replicative, it is replicative polymerase copy DNA. What I mean to say is that, let's say as a result of helicase, some portion of the DNA is being replicated. Okay, let's focus on a very small portion of the DNA. Let's say this is a double strand DNA and this is being maintained in a single strand state. Okay, let's say this is some DNA, and due to the action of helicase, one helicase is working over here, one helicase is working over here, and here the DNA at replication bubble, it is in a single strand state. Here the DNA is in a single strand state. If nothing is being done, then this replication bubble will again collapse. So there must be some kind of protein which keep or hold this DNA in a single strand state. This is done by these SSB or replication protein A. So they have some hydrophobic pocket, and due to which they bind to the DNA and stabilize the DNA in a single strand state, whether it is a leading template for leading strand or whether it is a template for lagging strand. At both the sides, what you'll find is these proteins which are binding over here. These proteins are referred to as SSB. So we can say that SSB, they are having some pocket by which they can bind to these nitrogenous base, do not allow them to interact with each other, they do not allow them to carry out an hydrogen bonding, so that replicative machinery, DNA polymerases, primerases, etc., they can come and can start the synthesis process. So the third protein what we have studied is the SSB, that is single stranded binding protein. Moving further, the fourth protein is, we have already mentioned about the initiator protein, we have already mentioned about the helicase. We need another protein that is referred to as helicase loader. That is, helicase itself is not having an ability that it can recognize an origin. The role of helicase loader in case of E. coli, that is in case of prokaryote, is by a protein referred to as DNA C, while in case of yeast, that is example of eukaryote, the same could be played by CDC6 and CDT1. They are regarded as a helicase loader. What I want to say, what I want to mean, or what I mean to say is that let's say this is some point from where the replication is going to start. Let's focus on a very small portion. This is the place from where the replication is going to be initiated in the prokaryotes, and this place is referred to as ORIC. Okay, this is the place from where the replication is going to start. This is ORIC. This ORIC, as already mentioned, that this ORIC, it is recognized by some protein which is having high affinity, and this protein is a DNA A protein, which is an initial protein. DNA A will see later on in its ATP bound form, it can recognize the ORIC. Now here a helicase must be recruited, but the helicase, that is DNA B, it itself not have an ability to interact either ORIC, or neither it is able to recognize a DNA A protein. But there is another protein referred to as DNA C. DNA C, it is having a affinity for a, so this is, you can say, DNA C, a helicase loader. It is having an affinity to recognize a DNA A. So we can say DNA C, it recognize DNA A and recruits DNA B, that is an helicase. Same is done in case of eukaryotes, that CDC6 and CDT1. Both of them, they recognize, or more accurately, they interact with ORC126 and recruit MCM227, which is basically a helicase. So the role of helicase loader in case of bacteria is performed by DNA C, and the role of a helicase loader in case of yeast or eukaryotes is performed by CDC6 and CDT1. So that is the helicase loader. Is it clear? So first protein what we have discussed is the initiator protein. Second, we have studied a helicase. We also talked about the polarity of helicase. Third, we have studied the activity of, you can say, SSP or replication protein A. Fourth protein what we have discussed so far is a helicase loader, because helicase themselves, they cannot interact with a origin or a initiator protein. So another protein come into play, and that is referred to as helicase loader. Then moving to further, that no DNA polymerase can initiate the replication process. So we need primase. Another protein which comes into play is the primase. The role of primase in case of E. coli, the role of the primase in case of E. coli is performed by protein referred to as DNA G, while the role of primase in case of yeast, or we can say higher organism, is performed by DNA polymerase alpha. That is one of the subunit of DNA polymerase alpha has a primase activity. So there is no separate primase in case of eukaryotes. One of the subunit of DNA polymerase alpha has itself has a primase activity. But what is the function of primase? Primase, it adds short oligonucleotide RNA primer. It could be from 2 nucleotide to 10 nucleotide. Most often it is around 5 nucleotide. Now what happens is that no DNA polymerase can initiate the replication process. So first a primase activity is necessary, and what primase do is that it first adds some RNA nucleotide, a short oligonucleotide. This could be represented like this. Now what these primase do is, let's say this is a template strand, and no DNA polymerase can initiate the replication, whether it is a leading strand or whether it is a lagging strand. Let's say if we represent it like this, This is 3 prime and this is 5 prime. Okay. No DNA polymerase can initiate the replication from the ends. First few nucleotides, they are added by a primase. This is 5 prime to 3 prime. And another important thing is that this is, here you utilize NTP. That is, this is an RNA. Instead of dNTP, you are using an NTP. RNA is being synthesized. Whether it is a leading strand or lagging strand, first few nucleotides, whether it is a leading strand or lagging strands, the first few nucleotides, they are added from 5 prime to 3 prime direction. They are added by a primase. So the role of primase is to synthesize the short primer, whether it is a leading strand or lagging strand. So what is primer? Primer is short ribonucleotide sequences, short RNA sequences which are added, and once this primer is being added, then further this primer could be extended. Then this primer or 3 prime strand of the primer could be extended by the DNA polymerase on this side or DNA polymerase on this side. Later on, DNA can be synthesized. Okay. So this is the DNA. And this DNA synthesis will occur, or it will be performed due to or activity of DNA polymerase. But now one question may come in your mind: why first we need this RNA primer, or why a DNA polymerase cannot initiate the reaction? The question is straightforward, that first few nucleotides, when they are added, proofreading activity is not possible. That is, if some wrong nucleotide is incorporated, it cannot be checked, because for proofreading activity, we will study later on, there must be some, we can say, template where a DNA polymerase backtrack. It can move in a back direction, but when 6 up to 5 to 6 nucleotides they are synthesized, all these nucleotides, these remain within the active site of enzyme cannot backtrack. So if some wrong nucleotide gets incorporated during the initiation process, they cannot be proofreadden by a DNA polymerase. So in evolution, it was decided that no DNA polymerase will initiate the reaction. The reaction is initiated by the primase, and primase, instead of adding a DNA sequences, it is adding an RNA sequences. And benefit of adding an RNA sequence is that again it could be recognized later on that this is the patch where proofreading activity has not been performed. So during the second round, when the RNA primer would be removed and gaps would be filled, during that particular period of time, proofreading activity would be performed. That we will still come to that particular point when we will remove the primer, and we will see that then DNA polymerase, they will able to perform a proofreading activity. So once the first few nucleotides they are being added, no proofreading activity is possible. But after 5 or 6 nucleotides they have been synthesized, now if a DNA polymerase it extends the reaction, it can carry out a proofreading activity. So the first initiation process or first synthesis of few nucleotide is performed by primase, and primase, instead of a deoxyribonucleotide, it add a ribonucleotide. So means it add a RNA. In other words, we can say primase is more like a Primase is like RNA polymerase. That is, primase, like an RNA polymerase, use DNA as a template, but this is the DNA which is being utilized as a template, but in front of the DNA, it is not synthesizing the DNA, it is adding the RNA. Is it clear? So this is the primase activity. Then moving further, that next enzyme what we are going to study is the major enzyme which is involved in replication, and the major enzyme which is involved in replication is the DNA polymerase. The important DNA polymerase in prokaryotes are DNA polymerase first, second, third, and we are also going to study DNA polymerase 4 and DNA polymerase 5. While eukaryotes, they are having variety of DNA polymerase, namely alpha, beta, gamma, delta, epsilon, micron, lambda, theta, kappa, etc. So we are going to study these DNA polymerase in the next class in a details. This was simply an introductory class about the molecular biology. So you should focus on these elementary points, that what we mean by replication, what are the stages where this replication is initiated. Then we must know the basic properties of replication, prokaryotes and eukaryotes, that it is a semi-conservative process, how it could be proven, why DNA replication is referred to as semi-discontinuous. The next point, you must know that DNA polymerase is a template dependent process, that should also be known to you. Okay, and we have also studied some of the important proteins which participate in a replication, like initiator protein. We have studied helicase, helicase loader. We have also studied the SSBs. We have also mentioned the role of primase. And in next class, we are going to study the DNA polymerases. So if you have any kind of problem, do send me as a message. I will try